Okay, so this is going to be the video for the Chapter 10 Mendelian Genetics chapter, and going to start off with kind of an assumption that you know most of the basics, like what alleles are, um, and phenotype versus genotype, which I'll talk about a little bit, and then the majority of this video will be about the Punnett squares. So let's start off with alleles. Really easy topic. Well, it can be a little confusing. There are a lot of terms like alleles, genes, and whatnot that can get intermixed and confused. But alleles basically um, a form of a gene that codes for a particular trait. So what do I mean when I say form? And that basically means, well, let's take a trait. Eye color is a trait. It's often confusing. People may think that I have brown eyes, so the trait is brown. But that's not true. The eye color is the trait. The trait that I actually have is the eye color, or my hair color, my skin color, and etc. And dimples and all that stuff. That's the trait. The form is the color of your eyes. So an allele basically tells you one allele is a different form. So I know that sounded a little confusing, but you'll see it most often like this. Maybe for eye color, we'll look at big B, little b. Well, in this scenario, when we're looking at eye color, big B is dominant, like always. The capitals are dominant, and it means brown eyes. And little b is recessive, and it means blue eyes. So what we're looking at here is two of the same letters, right? Because they code for the same trait. They're both for eye color. But they're different forms in the sense that one's brown and one's blue. One's dominant, one's recessive. And another way to know that they're different forms is that they're on different loci, or a different locus, which is a place or a point on the chromosome. So that's interesting. And so now I'll just get into quickly phenotype versus genotype, and this is probably the simplest concept in the chapter, but I'll explain it nonetheless. Alright. Phenotype, I like to think of the pH, the physical, because I think physical appearance, right? Because that's what it is. I can have the genotype big B, little b, and since brown is dominant, my phenotype, my physical appearance is brown. I have brown eyes, which is true. Genotype, you look at gen, which means beginning, and the prefix means beginning, gen like Genesis. So you can think of that as kind of like beginning for a human. And you think of the genes. Genes for the trait. So the genotype that I have would be big B, little b, because those are the genes, the alleles that I have for the phenotype of brown eyes. So now I'm going to clear this and look at Punnett squares, how we manipulate and cross different organisms using a little bit of math. So first, this is not going to be the best square for sure, but my penmanship on this tablet is not perfect. But first, basically, what you want to know is how to set this up first. And what's going to go on this side is going to be a gamete, gamete alleles from parent 1 and this side will be gamete alleles from, you guessed it, parent 2, right? Because basically what's happening here is you can say that up here is the sperm. We're going to call this the male, for the adult male. And here is the egg. And when we cross those two together, I'm doing this Punnet square work here, our result is fertilization. Fertilization. So, or a zygote that now carries whatever, depending on the probability of the outcomes here, like a 1 to 2 to 1 ratio. That's what you'll see when we do these monohybrid crosses. So now we'll look at actually doing some examples. So I'm going to assume that you know how to do a basic cross. Maybe if we're looking at that BB example, if I got one gamete or one gamete pair that kind of looked like that, and then another gamete pair, maybe from my mom, that was homozygous for brown eyes. Well, let's just do this. You got BB, or big B, big B, big B, little b, big B, big B, big B, little b. So you can see that here and here they're the same, and here they're the same. So basically it's a 50% chance that offspring is homo, meaning the same, homozygous for brown eyes. Homozygous brown. But now the real question is, what's the percentage chance that the offspring actually has the phenotype of brown eyes? And that's 100% chance, right? Because every single one of these, has the dominant brown allele. So they're all going to express brown no matter what, because it dominates over the blue here, the small blue. But it's 50% chance that the offspring will have a homozygous brown, meaning big B, big B. And then, of course, 50% chance it'll be heterozygous brown, big B, little b. So now we'll move into something a little more complicated. Still a monohybrid cross, but using sex-linked traits. And the one we're going to use specifically is hemophilia, because we did a homework assignment on it, and I just love that, and I'm sure we'll find something on it in the test, either in the form of multiple choice or a free response short answer question. So basically, this says that hemophilia is a sex-linked trait where the allele X, superscript capital H, is normal blood. So your platelets are doing the work they should be doing. Normal clotting, and the allele X, superscript lower h, is hemophilia. Hemophilia, which is a blood disorder. Hemo's blood, and philia is love. So philia like love or attraction. So it kind of means that you bleed too much, your platelets can't clot the blood flow after you get it cut. So you have to be very careful and whatnot. So it's definitely not a good thing to have. So it first says give the genotypes. So G types, all right, for a woman with normal blood clotting. A normal woman with father who had hemophilia. had hemophilia, and we want the genotype for this woman. So you have to kind of work backwards here, and you're going to have to draw her Punnett square. So first I'd draw the father, which father a male, so he's going to be an XY. So, and we know he had hemophilia, so the X has to be lowercase. So it's going to be X, lowercase h, and then just a Y. And then we'll do our little Punnett square. It's kind of small. And then for the mom, we were given no information. So it's best to assume that she did not have hemophilia. She had normal blood clotting. So at H capital, and then we'll cross it, and we get H capital, H lower, X H capital, X H lower, and then X H capital Y, X H capital Y. So now we look at this and we go back to the question. It says it's asking for a woman, right? So which of these are women? Well, not this one, not this one, these two, right? And so these the genotype of a woman who would have oh, sorry. I was totally thinking on the wrong track. I thought we were trying to find a woman with hemophilia. So we're trying to find a normal woman, I'm sorry, normal woman whose father had hemophilia. I confused myself by not writing the whole question down. But a normal woman, X capital H is dominant, right? So I don't know if I said that, but X capital H is dominant. So either of these would be a proper genotype for a normal woman. So you say that the genotype for her would be X capital H, X lowercase h. And you might say, well, couldn't it be X capital H, because that's a woman, XX, and she's normal blood clotting because it's two capital H's, there's no lowercase h. But to you, I'd say that would mean her father did not have hemophilia. You have to actually do the cross and see that the father had hemophilia that was expressed because the Y cannot compensate for this X with the hemophilia gene here. So it's important that you note that the father who had hemophilia is important because that gives us this lowercase h crossed with an uppercase H. So the woman did not have hemophilia. She had normal blood clotting. So the XH over here dominates over the X lowercase h, normal blood clotting. Now, a normal man whose father had hemophilia, so that's the same question, right? Exact same thing, but for a man. And we see, shouldn't have crossed that out over here, but over here, remember, it's going to be X capital H Y, because X capital H means that it's normal blood clotting and there's no lowercase h. Even if there were X, even if it was just completely random example, if the Y happened to have a lowercase h, which is not the scenario, can't happen, and it's not what's happening in this problem, but if the Y did have a lowercase h and also meant hemophilia, it wouldn't matter because the X uppercase H is dominant. So he'd still have normal blood clotting. But that's not what the problem is trying to tell us. So that was just kind of a bad example. So let's move on to some of the harder stuff. That was pretty easy. I did confuse myself in the beginning, but it was easy. Oops. So let's go here into the next question. It says, what is the probability that a mating between these two individuals will produce a child regardless of the sex that has hemophilia? So this is very easy. Monohybrid cross. I'll just go ahead and draw it. And remember our from the part 1, our woman with normal, her genotype was X uppercase H, X lowercase h, and our man with normal was X uppercase H, Y, right? Dominant. So let's cross those two. Oops. Okay, select my tool again. Oh, don't want to do that. Okay. Alright, it's kind of lopsided, but it's okay. Alright, so let's go ahead, and I like to do the male on top, and we're talking that these two people had a child, so we're trying to find the probability of their offspring having hemophilia. So this is our male on top, and our female on the side, XH, X lowercase h. So I'm going to do this in a different color. And we're trying to find a probability. So if we cross this first one, we get X uppercase H, X uppercase H. If we cross here, we get X uppercase H, Y. Here we get X uppercase H, X lowercase h. And here we get X lowercase h, Y. So now let's take a look at what these mean in terms of phenotypes. Do they express hemophilia or not? Does this express hemophilia? No. X uppercase H is normal. So 2X uppercase H's means it's a normal woman. So that's not what we're looking for. We're trying to look for something that has hemophilia, or regardless of sex. So we can eliminate that. Let's go down here. X uppercase H, Y. That's going to be a male, right? But we have an X uppercase H, so no, right? It's normal blood clotting. Okay, down here, X uppercase H, X lowercase h. People could get confused on this. You have a lowercase h, but you have to remember it's recessive. The normal blood clotting is dominant, so it's going to cover up. This X chromosome is essentially going to cover up this X chromosome and kind of say, oh, you don't exist, I'm dominant over you. This person, this woman does not have hemophilia, she has normal blood clotting. So we can eliminate that. And that leaves us with this. Well, let's look at it. We have an X lowercase h and a Y. So that's going to be a male. And this male does have hemophilia because we have the hemophilia here and then no X chromosome to cover it up. No X dominant chromosome for normal clotting to cover it up. So it's going to be a 1 in 4 or 25%. Right? That's pretty easy. So if this couple has a daughter, what is the probability that the daughter will be a carrier of the hemophilia trait? Well, we can use what we have here. I'll just redraw the square really quickly. Draw it a little bigger this time. I'm not going to put the traits up top and on the side. I'm just going to have what we had in there. XH, XH, XHY, XH, XH, XHY. So now, daughter. So which one are we looking at? We are looking at this column. We can completely disregard this stuff, right? Because they're XYs, they're male. So a carrier of the hemophilia trait. What you have to know is carrier does not equal expressed. So for all I know, I could be a carrier of the hemophilia trait, but I don't have hemophilia. Basically, that means I could, okay, that was a terrible example. I'm a boy, but let's say if I had a sister, my sister could carry the hemophilia trait, but not have hemophilia because she has another X chromosome to cover it up. So she can carry that X lowercase h, but she can have an X uppercase H to completely cover it up. But that still means that when my sister has kids or has offspring, she can pass down that X lowercase h. And so there's still going to be that 1 in 4 chance that we saw up here that one of her offspring, assuming that she mates with a male that looks like that, or sorry, a male that looks like that, there'll be a 25% chance that her offspring have hemophilia. So basically she could carry the X lowercase h, but it won't be expressed if she has an X uppercase h as well, because that X uppercase h is dominant and it will completely cover up the X lowercase h. So now We are looking at a sample set of 2, not 4 anymore, because we eliminated these. And out of 2, who's carrying an x lowercase h? Not this one. So this one. This woman is carrying an x lowercase h. So the probability is 1 in 2 or 50% that she's carrying. And again, neither of these daughters have hemophilia because they have a dominant X uppercase to cover up. In this case, it's homozygous, but in this case, this heterozygous, this dominant covers up the recessive hemophilic trait or hemophilic allele. And the last one, if this couple has a son, what is the probability that he will have hemophilia? So we can completely disregard this column and look here. This is, let me just rewrite that. These looked like X uppercase H, Y, and X lowercase h, Y. Sorry, lowercase h. So again, let's take a look. Completely disregard this, so it's going to be a fraction out of 2. And because the XY is the male. So if we have X uppercase H, Y, we have this allele for normal blood clotting. And then we have the Y chromosome, which doesn't have the gene for the hemophilia. So this we know very easily is going to be normal blood clotting. Still leaves us with this one, X lowercase h y. We know that this X lowercase h is the hemophilia allele. And so the next question we would ask is, okay, well, is there something else to cover it up? Is there a dominant allele? And no, because our only other allele is that Y chromosome, and it can't cover up an X chromosome. And so yes, this offspring would have hemophilia. So again, it'll be 1 in 2 or 50%. If this offspring were to be a female with X lowercase h, X uppercase H, they would be a carrier, but that wouldn't be expressed because this X covers up this X. This video is now like 23 minutes long, so I'm going to call it quits and then make another one on dihybrid crosses.